about somewhere around almost 1 million Daltons. The 4 rings in the middle are known as the 20s proteasome. So the whole thing is 26, inside is 20. Remember, S refers to sedimentation coefficient. So it's how quickly they sediment through some sort of a density gradient. It doesn't refer to molecular weight. It doesn't refer to molecular density at all. It's more shape and weight that is included in S. Anyway, these guys in the middle are 20s, and that 20s is about 750,000 Daltons. That's an average number. Inner rings here, all of these here are made of they're heptads, so that each ring has 7 proteins in it, and they're heteropeptads. Although even though they're heteropeptads, are related. The 2 outer guys are alpha rings, and the 2 inner guys are called the beta rings. And each of those, like the alpha ring here, they're not exactly the same, but they're all alpha. So they're in the same family, they do the same sort of a thing. Outer regions here are known as the cap, the 19S cap, that 19S cap. And the 19S cap, function of that cap is to regulate the entry of a protein into this region here. So this cap is one that recognizes the ubiquitination on a targeted protein and allows it to enter into one of these proteasomal structures. The whole function of this proteasome, by the way, is to grade ubiquitinated proteins. Here it shows you the E2 and E3. E3 is the ligase. It takes the ubiquitin off the carrier and adds it to the substrate of the offending protein. But this cap recognizes ubiquitinated proteins and allows them in there. And the function of this whole structure, right, is to allow proteasomal or protease digestion of an offending protein or the substrate without subjecting the rest of the cell to the proteases. These proteases are pretty strong, and so they're inside. The proteasome doesn't have a membrane around it. It's not a membrane structure. It's a multi-molecular structure. So the outer rings here, these alpha segments here, these 2 alpha segments, have the function of sort of unfolding the protein. So it takes this protein, and as it threads in, it kind of unfolds it and stretches it out. And the 2 inner guys, the beta subunits on the inside, are the guys that have proteolytic activity. The beta subunits have the proteolytic activity, and they actually digest the protein. And as I said before, they spit it out instead of about 7 or so. It's sort of interesting, too, that one of the ways that we recognize, we talked a little about antibodies and immunology, but one of the ways that we recognize foreign, right, is that cells in our body pick up foreign proteins and foreign materials, and they feed it into their proteasomes, and they break it up into these little 7 amino acid pieces, and then they display them on their surface. Right? They display it on the surface. And then those cells that have these things displayed on the surface, these T cells, go around and sort of test the beta cells, the ones that have the IgGs on the outside, right? So these tester cells have that foreign substance in a little 7 amino acid section on the surface of the cell, and they go around testing the beta cells, saying, which one of you guys has the IgG that can recognize this thing. And when they do, and they contact like that, they set off a clonal development of the beta cells, and it expands it. But because there's 7 amino acid segments in all of these proteins, right, there's a whole bunch of them, it could identify different beta cells that have a different receptor, essentially, for the antigen. These little amino acid pieces are called antigens. They test the beta cells. And when the antibody fits the antigen, you get a signal. Well, there's a lot of antigens. You can activate many different kinds of beta cells, and in the process, you make lots of different antibodies, each of them to one of these little 7 amino acid pieces, right? So because of that, you can do several beta clones, beta lymphocyte clones, making different antibodies that recognize the same protein, different antigens of it. And that's why we call antibodies polyclonal, because they're polyclonal. There's many beta clones that are induced by these little 7 amino acid pieces of that same peptide, the antigen. Yes? So these proteasomes are extremely important also for recognizing foreign material and setting up antibody responses. Okay, does anybody have any question about proteasomes? So what we said essentially is that proteins in the cell, right, there are native proteins that are functional, but some of those are misfolded. And as I said before, there are certain proteins that the cell just have a heck of a time folding properly. But there's two pathways that can go in. The first preferred pathway in eukaryotic cells is at least to go through this process here involving these chaperones, right, that feed the protein in, allow that protein to breathe and refold, and hopefully become a native protein. Remember here, the rings at the top, you can see the protein's actually bound to it. Hydrophobic reaches that protein, bind to it, and then when ATP is there, it kind of releases it and lets that protein refold and rebreathe. The other pathway is destruction. One of these proteasomes, same thing, it has this inner core like that. Interesting that they're both made up of sets of rings like that. It sort of suggests that they evolved similarly. The top two rings actually do the same thing. This guy's unfolding proteins that are, hydrophobic regions interacting, and these alpha rings on these guys unfold that protein so that they interact, or they can be worked on. This one has proteases, this one allows them to refold again. But it goes either way, and there's sort of a balance between that and the cell to keep the cell cleaned up. So what happens if we don't clean up those degraded things, those aggregated proteins, those proteins that are not fixed correctly? I said before, they can cause problems in cells. Who knows what this is a picture of? Does anybody recognize that at all? Off the top. It's like 100 other different electron micrographs. Who said it? Alzheimer's, right? So this is a picture of the cerebral cortex in somebody's brain, and all of these little brown things, these big brown things, are what we call the plaques. So these are plaques in the cortex, the cerebral cortex, and when you have Alzheimer's disease. And these plaques, as it says here, are made up primarily of a protein that's called Biochem. It's not a protein, it's a peptide. What's the difference between a peptide and a protein? Protein sort of is a peptide in a way. Normally we refer to a peptide as a sort of a fragment of a protein. So this thing only has 42 amino acids. With a peptide suggests that it's either a fragment or something, and it is. So here's how it works. The beta amyloid peptide is derived from a large protein that's called the amyloid precursor protein, APP. And APP has about 750 amino acids. It varies between 756 and a few less, and we're not sure exactly why it's made in different sizes, but it is. What we do know is it's a class 1 protein, which means that it has a single alpha helix going through the membrane. Remember what we talked about? If you look at this is an alpha helix going through the membrane, and we said it took about 23 amino acids to span the membrane or so. If you count there ought to be 23 or close to that amino acids in this region that span the membrane. They're very hydrophobic. They exist in the lipids. If the hydrophobicity went any longer, it would not be able to bury itself in the lipid. Right? If it was any smaller, and these were charged residues on either of these sides here, it also couldn't exist in the membrane. So it has to be 23 or so that fit in the membrane like that. Why does an alpha helix go through the membrane? Did we talk about that? Why does an alpha helix go through the membrane? If these are hydrophobic like this, right, they're all hydrophobic residues, why can't they just stretch out and take whatever it is, 1, 2, 3, 4, 5, 6 in there, whatever, 8 of those to go through the membrane? Why does it have to be a helix? And it is. It's always a helix in the membrane. Why? Who can tell me? Well, we can't have a charge in that membrane, right? That's why these are all hydrophobic. No positively charged or negatively charged amino acids can exist in that environment. What about the peptide bond? Remember what the peptide bond is? Carboxy to amino. Right? So you have that carboxy that has a charge on it. You have the amino group that has a charge on it. So the peptide bond itself is charged. It can't go through the membrane. Right? But if you make an alpha helix, and I'm just going to do this briefly for you, I won't go fancy. But if you have an alpha helix like this, right, and you have an amino acid here that has a carboxy oxygen, and 4 aminos up here, amino acids here, which is about how many it takes to get right here. You have that amino, did I say carboxy oxygen? every fourth amino acid of oxygen and the amino nitrogen form a hydrogen bond and it neutralizes it. So in a helix like that, all of the charges in the backbone that's due to that peptide bond is all neutralized. So now, this helix has no charge and it can go through the membrane. So everything that goes through the membrane has to have no charge, has to be hydrophobic residues, and so by forming a helix, it can do that. So this amyloid precursor protein has a single helix that goes through the membrane. It's in normal cells. This is a protein that we all make. We make it in all of our cells. And it's processed in cells. And it can be processed in 3 ways. It can be cut at 3 different sites, which is what the scissors indicate. And the enzymes that make the cut have been termed secretases. And the reason is, right, if this is sitting in the membrane, if you make a cut here, then this part of the protein is secreted, essentially, away from the cell. If you cut here, this part's secreted away from the cell. If you cut here, this part's secreted into the cell, kind of. So these enzymes are called secretases. They're called alpha, beta, and gamma secretase. So they're normally processed. We don't know exactly what the functions of these guys are, of this protein is, or the processing is. We know more and more that this protein is very important, at least in nerve cells, for neural maintenance. So neurons, in order to survive once they've gone to their target cell, their target tissue, they have to receive a maintenance factor, which keeps them there and keeps them active. And we think this is involved somehow in that maintenance process, but we're not exactly sure how. We also think, or some people, not we, but some people think that this portion here that's in the cytosol, these dots indicate it's more. There's about 100 amino acids inside. From here, it's like 600 and something. I don't remember what. But these 100 or so amino acids, when they're released into the cytosol, gain access to the nucleus where they function as transcription factors. So some people think this is a transcription factor. Some people think that this part that's secreted from cells when it's cleaved here can actually bind to receptors on other cells. Right? There's a receptor for it, and that maybe has something to do with the maintenance. But bottom line is no one knows exactly what any of these do, and all of this is sort of still testing stage. This is a diagram that just sort of shows you the same thing. This is the same protein here. That's ATP. Here's the A beta portion. Remember that beta amyloid peptide is about 40 to 40